Hey there, welcome to my channel. I'm here, Brew, and today we're going to talk about a virus. Well, actually we're going to talk about the history of viruses, and for that we are going to go back quite a bit, all the way to the 18th century. So sit tight and let's do this. The first virus to be discovered was smallpox. This happened in the year 1796. It was discovered by Edward Jenner. He observed that milkmaids who had contracted cowpox, which is a mild disease, were immune to smallpox. Evolved. And because of this observation, he then did an experiment where he inoculated a boy with cowpox and then exposed him to smallpox. And it turned out that the boy did not get sick with smallpox. So that was our very first vaccine. And ever since then, we have been trying to do the same thing with other diseases. In the early 20th century, we were able to identify two types of viruses based on how they are transmitted, and these are still valid today. So the first one is called an arbovir. For example, we have something called antigenic drift, which is when a virus changes so much that our immune system can no longer recognize it. One of these viruses is called Ebola, and this virus causes a hemorrhagic fever. And the best way to grow viruses is in chicken egg. And that's what we did during the 1940s and 1950s, and we were able to grow poliovirus in chicken eggs. And this allowed us to develop a vaccine for polio. However, not all viruses can be grown in chicken egg. For example, we cannot grow HIV in chicken egg. So instead, we have to grow HIV in human cells. So it causes bleeding inside and outside of the body. And it has a mortality rate of up to 90%. So it is extremely deadly. There is another virus called Marburg, and this one is similar to Ebola. It is also a hemorrhagic fever virus. It causes bleeding inside and outside of the body. And it has a mortality rate of up to 30%. Then we have Lassa fever virus. This one causes fever, bleeding, and a rash. And it has a mortality rate of up to 15%. And then we have Crimean Congo, hemorrhagic fever virus. And this one also causes fever, bleeding, and a rash. And it has a mortality rate of up to 30%. And this is what happens with the flu virus every single year. That's why we have to make a new flu vaccine every year. Now, sometimes these changes can be even more significant. And if a virus changes enough, it can even jump species. B are just genetic material inside of a protein shell. Please hit the like button. Also, remember to subscribe and share this video with your friends. And I'll see you next time. And because they are so small, they cannot be seen with a regular light microscope. They can only be seen with an electron microscope, and that's called a pandemic blurs. And unfortunately, we have been dealing with a pandemic for the last couple of years. These are viruses that are transmitted by others. So we're talking about things like mosquitoes or ticks, or maybe even fleas. And the second one is a vector-borne virus. And these are viruses that are transmitted by an animal, usually a vertebrate, so something like a rodent or a monkey. And we know that some of these viruses can cause really severe diseases. For example, we have HIV, which is a human immunodeficiency virus. We also have influenza, or the flu virus. We also have hepatitis B, and we have polio. Unfortunately, thanks to vaccination program, Many of these diseases have become much less common than they used to be. For example, in the United States, we used to have thousands of cases of polio every single year. But now we only have a handful of cases. And those are mostly in people who are not vaccinated. Now, one thing to keep in mind is that viruses change over time. They evolve. And sometimes these changes can be really significant. Now, even though we have been able to develop vaccines for some viruses, there are still some viruses that we have not been able to find a vaccine for. Profound. The study of viruses is called virology. And in order to study viruses, we need to grow them in a lab. One of the challenges of studying viruses is that they are so tiny. They are about 100 times smaller than a bacteria, and they are not cells. I hope you enjoyed learning about the history of viruses. Thank you for watching.